Hi, everyone. My name is Edmika Rodriguez, a senior biochemist here at Humboldt State University in Arcata, California. And today I'll be talking about adeno-associated virus modification using leucine zippers. The paper is called Site-Specific Post-Translational Surface Modification of Adeno-Associated Virus Vectors using Leucine Zippers. The reason I chose this paper is because my goal in life is to bring happiness to those near me, around me, that I'm eventually going to meet, hopefully, all throughout my life, as well as everyone everywhere. Um, I wanted to become a biochemist to work with both proteins and diseases, just because, like I said, my goal in life is to bring happiness to those around me. And the only way I could think so of doing that was to make their loved ones live a longer, healthier life. Especially in times right now with the whole pandemic happening. And even before the pandemic happening, about 1.4 million people died and have died from either heart disease, cancer, chronic lower respiratory diseases, influenza, and so much more. This research that I was just talking about was conducted in the US in 2019. And adenovirus is one form of trying to solve these problems and helping them. So, and hopefully one day I'd be able to work with this and, bring smiles and happiness to the world. So before we get to the whole nitty gritty and the good stuff of this presentation today, I wanted to go over some abbreviations that are going to be seen in the presentation as well as heard. So AAV would be adeno-associated virus, which is kind of like a cousin of adenovirus, a synthetic cell. qPCR, quantitative polymerase chain reaction. Also, I'm very sorry if I mispronounce any of the words that I will be using during this presentation. The next abbreviation is LZ, the leucine zipper. Those are the proteins that were attached to the capsids. The next one will be EK, enterokinase, which is a buffer that will be used throughout the experiment. And finally, MOI, which is the multiplicity of infection. So this is the ratio of agent to infection target. So what is AAV and why is it important? Many of you might be asking that, and luckily you got the right man for the job. So while adenovirus is a synthetic cell, like I had mentioned before, that is perfect for gene therapy due to their viral vectors, which allow them to penetrate a cell membrane easier. Which is a way to deliver genetic material into cells. This allows for viruses to attack the host with the ability to do so without affecting or changing the genome or the genetic sequence of it. In other words, in simpler terms, this synthetic cell was designed to deliver new modified DNA to target cells. One way that it's used, like I had said before in gene therapy, is seen in figure 1. This figure was taken from US National Library of Medicine, where a new fully assembled capsid with a modified DNA is prepared and enters the cell membrane. Upon entry, the structure slowly begins to break due to the acidity increasing to the point where it's necessary for it to attach to the nuclear pore before it completely breaks. Upon attachment, the new modified DNA gets to deliver itself into the nucleus, where it becomes an mRNA, and it is able to infect and become a blueprint for protein production throughout the whole process. And figure 2 down below, it shows the different types of viruses and corresponding uses of each one. The virus specifically in this research is going to be AAV9, which you could see in the list is helpful for hemophilia, CT, DMD, muscle tissue, heart tissue, as well as CNS. These are just some of the AAVs that are currently being used, but there's always room for improvement, as well as new ways and new modifications to do new research. So I just want to quickly go through what is an adenovirus made of? Like, what does it consist of? So adenovirus could be made up of three things: a fiber, a penton base, and a capsid. Specifically for the fiber, it could be broken down to two parts: the knob and the shaft. These figures to the right were both received from the Protein Data Bank 101. They both are related to adenovirus. It was protein of the month. So yeah, going back to this figure, upon reading what that adenovirus was about, the fiber specifically contains a carboxyl terminal cell attachment with a beta variation of glycine or proline, as well as hydrophobic residues that allow it to penetrate a cell membrane and deliver the new modified DNA. The fiber is able to be held onto the capsid by the penton-based proteins. And finally, the final piece of the whole adenovirus is the capsid. The capsid outer layer consists of hexon proteins and minor proteins. And for the core, which is all this inside out, is a mixture of minor proteins and new modified double-stranded DNA, which can be found in the very, very center. So now that you know what adenovirus and AAV may be used for and the different types of things they're used for, as well as what they're somewhat made of, I want to go through what are the differences between AAV and adenovirus. So some of the differences, for starters, is that the sizes of each one differ. For adenovirus, it's roughly about 90 to 100 nanometers. And for AAV, it could be around 25 nanometers. The difference of sizes causes an even bigger gap with each of their packing capacities, with adenovirus having a packing capacity of roughly about 8.5 kb, and for AAV having a capacity of about roughly 4.5 kb. Just while going off of the whole capacity, they also have differentiation between protein expression, with adenovirus having a high expression and AAV having a very low expression, as well as their response to immune cells, with adenovirus having a high one and AAV having a very low one. Another way to distinguish them are the safety between both and when you're using them for experiments. But despite those whole list of the way to describe each one differently, one of the major ways you could differentiate between the both are the way they penetrate cell membranes. So figure to the left, figure 4, part A expresses a regular adenovirus, and the way they enter a cell membrane, they do so by a method called CAR. CAR is a Coxsackie and adenovirus receptor. And for B, that's an associated adenovirus, AAV. And that one is cell penetrated by heparan sulfate protogline receptor. So since we were able to see many ways to distinguish the both, I wanted to also show you the atomic structure of an AAV. We've seen an adenovirus one in the previous slide. For an AAV, figure 5 up here expresses one that I also found in the protein data bank. This just shows how the way they're different. We see that the adenovirus has the fibers. AAVs tend to not have fibers, but they also come in different forms. Some do have fibers, some don't. It just depends on the person conducting experiment of how they want to go about it. For figure 5 specifically, I know that it is a AAV2 virus. So AAV2, if you remember back from the list, it helps in kidney tissue, liver tissue, CNS, and much, much more. Right below that is a picture that somewhat gives a quick representation of what was being conducted for this research using modified AAV capsids with leucine zippers and a loop conformation went under interkinase digestion to linearize the looped fibers. And after that, the newly linearized leucine zippers were then binded with peptides and named Velcro AAVs. And that's how the whole research was done. But how did it begin? What were the steps first taken? So the first step taken when starting the research was to first assemble the newly modified capsid, Velcro AAV. The modification process began with AAV9 gene cap. With that cap, we were able to attach proteins and peptides to the surface using ALZ coiled coil heterization. I mean heterodimerization. So using the AAV9 capsid gene, we took out 3 samples, VP1 at subunit 1, VP2 at subunit 138, and VP3 at subunit 203. For VP2, E3, and K3, the researchers specifically looked at the residue site G453 because past research previously demonstrated that the insertion there was the most tolerated and accepting, of course. The protein inserted consisted about 21 residues called 3-heptad, and the heterodimer consisted of both K3 and E3 with nitrogen and carbon terminal groups attached to the capsid. And this could be seen in the reference I'm about to show you, which was taken from this research. And it is called coiled peptide linkers. So in this reference number 6, we could see that E3 is in blue and K3 is in green. Besides adding this protein, we also added spacers that could be seen in red at sites subunits 453 for both K3 and E3. And we also included interkinase cleavage in orange. We added both of these to help it make it more flexible in the insertions, as well as facilitating folding and virus assembly. I meant assemble. So after each subunit was pulled from AAV9 to create our 3 samples, some of the codons from each sample were also removed through site-directed mutagenesis, also known as SDM, for all the samples. This was done to remove the weak codons that started each sample. So specifically for VP1, they included VP3 as well as a new set of codon for their starter. For VP2, E3, and K3, they removed the starter codons as well and replaced them with new codons. So to make sure that the modified capsids were finally assembled and stable, they use plasmid transfection followed by purification and then finally quantified. This was done to, double check that everything was all set and prepared. The plasmid specifically consisted of adenoviral helper genes, as well as VP13 and VP2LZ, which are the leucine zipper proteins. About 48 hours after the whole plasmid part of that experiment happened, the samples were lysed through freeze thaws and treated with benzonase and centrifuged to remove excess nucleic acids. And finally, the supernatant, which was about 40% iodexanol layer, was collected and concentrated with granite buffer and quantified. The quantification was done using qPCR. For the PCR samples and the data collected, I'm about to show you in the next slide, were denatured in NaOH to release genomes and neutralized using HCl. For the final step for this whole prepared part of the experiment, I find really interesting because the samples were diluted in salmon sperm and pure water. The data recorded and seen in part A is the viral genomes per milliliter, which is the concentration for the samples. The controlled sample was AAV9 and samples E3 and K3. The result of this graph was that about roughly 25% of the unmodified AAV9 titers formed E3 and K3 vectors, and the inserts at that same site have not impacted the whole assemble of capsid and genome packaging, as well as accept the different sizes and different sequence of insertion depend on the tolerance of each capsid. So with n equaling 3 for the amount of independent viruses prepped, the SEM, which is standard error of the mean, was calculated and shown by the error bars attached to each sample data in part A. For part B, it's a Western plot created with samples that were treated with either enterkinase or sham. They were then denatured. After that, they were attached to B1 antibodies, lamin, and was used to recognize epitope present in the C termini. So that resulted in graph B resulted in both K3 and E3 containing modified VP3 proteins, despite being modified with VP2LZ proteins. And that could be seen by the faint little strip right here. They were also able to conclude that the C-terminal cleavage was successful due to the bands disappearing after this. This was due to the digestion of EK, with causing them to see a KDA mass of 32. So after this whole characterization thing, they asked themselves, how does EK or S sham affect the capsid structural integrity? To answer that question, researchers had to dilute the virus samples 1/10 in endo buffer, which help isolate the plasmid DNA, as well as genomes that remained were quantified through qPCR once again, and that is seen in the next slide. So in doing so, that allowed for the calculation of genomic protection between benase or sham treated samples. These were important due to the reaction causing nucleus activity. The end result of that was that E3 and K3 exhibiting about 75 to 100% genomic protection, similar to the control group AAV9, despite the whole EK treatment that it underwent. As well as I wanted to mention that the error bars that is seen in the graph are once again associated with SEM. So to do a quick overview of what this graph and data means is that the EK digestion of LZ linearization does not affect the whole capsid structure. So now that we know the whole structure is stable and everything's working just fine so far, how can we tell if our Velcro AAVs actually binded to their complementary LZ protein successfully? So researchers thought of using nickel affinity chromatography. They designed LZ peptides with C-terminal His6 tags, which allow them to see the successful binding of their sample. The samples were roughly about 3 × 10⁹ VG viral genome, which is about a weight. Those Velcro AAV were then diluted in 120 microliter of binding buffer with LZ peptides. That resulted in E3 being isolated with K3 His6 peptide and K3 being incubated and isolated with E3 His6 peptide. And as we could see in our graphs, a set of 4 solutions were collected with different concentrations of imidazole and tested with a qPCR. Titers were normalized to total amount of recovered viruses from load, wash, and elution. So the results indicated that E3, which is graph A, incubated with about 300,000 X of K3 His6 peptide was roughly 80% binded to the column, and about 30,000 X, which is somewhere around here, a ratio resulted to about 20% binding. As for the lower concentrations, not being induced to column binding, so they just, flowed right through. K3, on the other hand, which is graph B at the bottom, didn't exhibit any column binding throughout the whole elution process. The controlled AAV, which is in black for both graphs, on the other hand, did not show the desired column binding, and it was due to the interaction of the LZ being joined by 2 different subunits. And just to show a quick example of what a column looks like, currently in my biochemistry class run by Jenny Capuccio, we were able to make columns. I don't specifically remember what this column was used for at the moment, but this is what a standard column looks like. And we just take fractions of our wash, which is represented by the W, our flow through, which is represented by the S in the graph, and our elution fractions, which is E. So in conclusion, the attachment of peptides to AAV was successful with LZ adapters and demonstrated the ability to do so with complementary adapters. Intact Velcro AAV capsids were also able to stay intact depending on the insertion and cell type. They were also able to see that E3 is able to bind to complementary K3 peptides, as well as the promise of AAV peptide attachments being displayed. Future experiments may find optimization of the number of LZ incorporated in the capsid, resulting in higher levels, hopefully, of peptide and protein display, as well as reduced vector prediction yields. This may be done by adjusting plasmid ratio of VP2-LZ and VP1-3 used in vector prediction, or using stronger start codons at VP2 in the VP2 LZ plasmid that would help reduce VP3 LZ expressions. And in doing so with that, reduces LZ containing subunits. So the potential of functionalization with antibodies and enzymes is there, as well as using Velcro AAV for LZ binding to display multiple ligands, which help in targeting and delivery to become more successful. And with that, I would like to conclude my presentation. Thank you for your time and look forward to seeing you and the questions you have for me. Have a nice evening.